Have a mic. It's us against you. I'm gonna pass the mic around and ask everybody to introduce themselves. And if you're here because you have a family member with MPAN, I would ask that you just give us maybe 2 or 3 sentences about each of your loved ones with MPAN. Just tell us a little bit about your family, because that might help us kind of in how we can help you. So I'm Susan Hayflick. I'm a medical geneticist, a physician scientist. I'm not a neurologist. I'm not very good with medicines. That's not my strength. But I've been working on the NBI disorders for a long time, 30 years, and taken an interest in every one of them. And I think we're here with only MPAN families, no FAN families, so we're not going to talk about FAN here, but I'll talk a little bit about it. I'm doing a research overview on Saturday, and so I will be talking a little bit about what I know about what's going on worldwide with MPAN research on Saturday, and also FAN on Saturday and others. But I think we're here to primarily focus on clinical discussions, but the floor is pretty much open as far as I'm concerned.
Okay, I'm Penny Hogarth. I'm a neurologist, also from Oregon. I think I know many people here. So I've been working with Susan and with the NBI disorder since I came to OHSU, which is now 23 years ago. And so that's who I am. So.
Jim Bourgeois, I'm a psychiatry professor at UC Davis, but for our purposes here, I'm a parent of a 23-year-old MPAN child, child Germaine.
I'm Allison Gregory. I think you all know me. I'm a genetic counselor. I've been doing this with these guys since 2002. Yeah, that's it. Do you want to introduce yourself real quick?
I'm Lakin. I'm a second-year medical student also from Oregon, and I have not been doing this a long time. This is my first MBIA conference.
Hooking him in.
I'm Kathleen Ayers, a clinical psychologist, and Dr. Bourgeois is my husband. He's already told you about our daughter.
I'm Peggy Wicker. I'm a grandma, a mother, and a wife. James, I have a picture right now. He just turned 33 years old. He has MPAN. I'm sorry, you could pass it. Yeah, he just turned 33. I forgot what I was supposed to say. Yes, he lives in a facility, Hattie Larlam. Yeah, he needs, at this point, he needs 24-hour care. He's 4.5 years ago, he had ended up with the RSV virus, and then he ended up with pneumonia and his lungs collapsed. So then to save his life, we opted for a trach. So he's been on a trach and a feeding tube for probably 8 years. But yeah, he can do facial expressions. Can't really move anymore, but okay, I'm gonna pass this on.
I'm Michael Wickert. I'm dad and husband, and there's Jim, and then there's Cameron too, and Cameron's big brother, and he's in Philadelphia with Brittany Aviana Taylor, which is pretty awesome because he named her after James Taylor. So so thank you all for being here and doing what you all are doing, because it would be, as we talked about, first timers, it's a very lonely trip the first time. So thank you.
Good morning. Cheryl Amos. I have a 42-year-old daughter with MPAN. I had 2 other children with MPAN that already passed on. But Barbara had a slower progression than Becky anyway. A blood clot got bruised, so I don't know how that would have progressed. But Barbara is lovely, and I'm going to be sharing my story. You'll find out more about me.
chime in. She has two adorable grandchildren.
Oh,
You better both grab the
totally
pictures.
forgot
Yeah.
about
Can't forget.
Ben. Ben called me because I sent him a Word doc copy of my speech, and he said, "Mom, I'm so proud of you. You go." Of course, I will. I've shown everybody here, I think, my grandkids already anyway.
Good morning. My name is Roberta Leonardi. I'm the new member of the Scientific and Medical Advisory Board. I'm very familiar with PCAN, but I'm here to learn more about the other diseases in the under the NBIA umbrella.
Hi, I'm Jenny Wilson. I'm a pediatric neurologist at OHSU, along with these guys, and I've been involved with the NBIA disorders for, I think, 7-ish years.
Hi, I'm Nivi Thakur. I'm from the Children's Hospital of Philadelphia. I'm a movement disorder specialist, and I think, actually my roots are here in Houston, so I've been able to meet so many of the families that I started to get to know, but I think my journey's continuing up there.
So I think some questions were submitted. Okay, so maybe Allison can kind of lead us from here.
Sorry, I'm balancing two meetings here. So you all had the opportunity to submit questions, and we can start with these, but obviously we're having an interactive discussion here, so we'll just start off. And maybe we can just, whoever feels they can answer can field these. The first one was, what are recent advancements in understanding the natural history of MPAN?
So I can sort of take a start at that. I was talking a little bit about that earlier this morning. So natural history is how the disease progresses over time. It's a really important baseline for the community to understand, and it's one of the most important data sets for therapeutics development. The FDA and the European equivalent will basically require some baseline information. So MPAN is really variable. You all have adult children, but there are people with more aggressive forms, those leading to a more rapid progression and earlier death, and actually later as well. So there's quite a wide spectrum for MPAN, and it's important to capture that whole spectrum. Unlike most of the other NBIA genes, the MPAN gene was found by the Germans in collaboration with the Poles, because there's a very large population in Poland and Germany in Eastern Europe, due to this founder idea, somebody with a mutation and a relatively kind of a relatively small community. So the gene was found in Germany, and we have, even though we take care of a lot of MPAN families, we have sort of largely respected the German and Polish leadership in this area. And so they have organized a natural history study for MPAN. I was going to ask Allison if she knows anything about that natural history study, and whether it's also got an English language arm to it, because I think it's important that the American families and other English-speaking families are able to participate. But in fairness, we don't have a Polish arm to the BPAN natural history study, and so they may in fact not. But I'll ask Allison to respond to that.
I don't think is it on? Yeah, it is on. I don't know anything about that. I haven't heard from them. I did email with the family leader of the Polish family group recently about something else, but that didn't come up. But the one thing I would say is, because of the MPAN trajectory and the way that it is sort of more slowly progressive, and your kids are, getting to be at older ages now, we've known you families in particular for a really long time. So I think we have good natural history information sitting in our database, including medical records, and even, when we've seen your kids and examined them at past meetings. So we'd have something, if there were an opportunity, I think there's stuff to be contributed from our group on a lot of, and especially with, a couple of the papers we've done too. We've got 50-ish cases probably in our database.
Yeah, good point. So we could potentially put something together as well, yeah.
I can't speak to the natural history studies in Poland, but we were doing monthly Zoom meetings with the MPAN group until about a year ago. And the Polish researcher, one of them was on the line, they were doing, I think about a year ago, they were moving into a study with diet and supplements. But that's all I've heard since then.
Yeah.
So we know the Carter family as well, and I don't remember what version they have, but they have been very diligent with the food, you know, and that, and those kids are doing great, right? And the other thing I would ask is that, you know, if we like need to sign a waiver or something to share that data that you've collected, cool, let's do it. And the other thing is, can you forward that Polish, the history, you know, email or whatever? And we're not good at Zoom, we're not good at technology, but if there's a way that we can get connected, is what I'm trying to say.
If you're here on Saturday, I'll actually have a screenshot of the Polish NBIA if you just Google NBIA Polska. And it says there's an English language, you know, you can usually choose your language. It says there's an English, I chose English and everything was still in Polish. But this is a good opportunity for you to work on your Polish.
Well, and as far as the sharing goes, thank you. I think I can safely say that all three of your families are consented in our repository, have been for a really long time, which means you've already agreed that we can share what's called de-identified information. We just scrub off names and use a number. So that's in place, but if we needed more records, you know, like an update, a lot's happened with James, for example, in the last 8 years that we haven't been as involved in, so that might be useful at some point. Yeah. So the other two questions, one of the other questions was asking about a research update, and I don't know if we just want to save that for Saturday for that session, as particularly what's been going on new in the last 2 or 3 years was the question.
Yeah, I'm happy to do it here as well. So I was talking a little bit earlier this morning. The challenge in the MPAN community continues to be we don't know what the protein does. So in all the other NBI disorders, we either know or think we know or imagine that we know. And in MPAN, we're kind of still floating in the clouds. We really, really don't know. We know that we know some of the downstream biology loosely, but we don't know. It's like kind of the downstream effects of a dysfunctional protein. But there's really important, very fundamental, basic, foundational science that needs to be done. And it's not that nobody's tried. A lot of people have tried, and they have not made inroads. We, the community, have taken multiple different angles to try and get at it. There's been a structural biology program. Sometimes the structure of a protein can teach us about its function. Not a lot of progress that I've been able to glean from that. No publications yet from that group. And so I think we need kind of new eyes on the problem, and there is an interest now by a group in Italy in trying to gain a foothold of understanding on the MPAN protein function. There are some models, so there's fruit flies with MPAN, and I think you'll be hearing at the Saturday research talks, you'll be hearing a little bit there. And then in Germany, Dr. Ayuso will also be talking about some of their models and their work, including human cell work. And the group in Germany works with others in Europe. They work well with the Poles and they work well with the Italians. And so, you know, these are not groups competing. These are actually, there's really good collaborative spirit in the MPAN scientific community because everybody's doing the same thing, walking around scratching their heads. But that's important that there's collaboration and resource sharing. So the most fundamental work that still needs to be done is, I hope, somebody's going to take a new run at it with some new models and new partners, and that's the best I think that can be done. Not all treatments, not all therapeutics depend on knowing the function of a protein. Gene therapy is the main one, where if there's a mutation, a misspelling in a gene, and there's sort of the idea that if you can deliver the correctly spelled gene to the cells, particularly cells in the brain for MPAN, that you might be able to impact the way that the disease progresses. The challenges in MPAN, so MPAN gene is a very small gene, which is good. Like it's literally it's a short sentence, not 3 pages. It's a short sentence. So the cargo that needs to be delivered, the gene that needs to be delivered, is pretty small, and that's actually advantageous from a gene therapy standpoint. The challenge is that MPAN doesn't sit in just one tiny part of the brain like PCAN. PCAN sits in a very small part of the brain. MPAN sits everywhere in the brain and beyond, but really extensively. And so it wouldn't be, I think, wise to try and target a small subset of brain cells. I think you would need to hit the whole brain, and that requires a large viral load and a high efficiency sort of viral delivery system. And there's a great deal of work because this is a problem for many of the genetic disorders, whole brain, kind of hard to reach. It's a problem that's being worked on by a lot of companies and a lot of academic scientists. It's a very high priority problem, but it's not one that's been solved yet. So, and the question is for an adult, at various stages in their disease, what are the risks and what are the potential benefits? I think particularly for brain disorders, long-standing brain disorders, it's probably not realistic to expect, even with the best gene therapy, to expect any kind of reversal in the disease. I don't think that's consistent with what we know about the protein aggregation in the disease. In fact, it may be that gene therapy, once there's enough of the disease-associated protein called synuclein, that it may be that sort of horse has left the barn, as they say, that the process, the synuclein aggregation process, is running on its own, and even swapping a new gene in may or may not impede that process. What happens is that protein glumps together, and as it glumps together, it sort of becomes more and more attractive to more glumping, and that causes, kind of dysfunction in a lot of the processes in the cell. Nevertheless, I think gene therapy, because it doesn't rely on that knowledge of the protein function, I think gene therapy for earlier disease and maybe for young people may hold some promise. I'm not aware that anybody's working on gene therapy. They may be. I'm just not aware that anybody is for MPAN. And I think that the Polish group is working on a variety of either botanical or what we would call over-the-counter supplements to see if they influence the natural history of MPAN. My expectation is that is not to learn the results of their studies from what they say on the website, but to actually wait until they have published a peer-reviewed paper. So in academics, the way that new knowledge is presented in a generally accepted way is not me getting on Facebook and saying what's true. It's me writing down everything we did and what our results were and how I interpreted that, and then having other scientists look at it and critique it and say, this is bunk. And this will not be in the public domain. You may not publish this. Or they will say, you have to change this or do 2 more experiments. It sets a level of rigor that is really important. And so I think it's particularly important because I've gotten some kind of some wow sort of statements from some of my colleagues in Poland about the results that they're getting for some of the work that they're doing. And I'm hopeful, but I'm a little skeptical. And so I want to see independent review that is confirming what they're saying. And actually it's with a BPAN compound that they're reporting anecdotally. They're reporting kind of wow results that I'm very skeptical of. Yeah.
One of the comments. So something that you'll hear me harp on a little bit as we talk today is something that is, it's not unique to MPAN, but I think is a potential big advantage to MPAN in our being able to move the needle towards treatments, is the ways in which MPAN is related to Parkinson's disease. So most of the NBIA disorders have some connection to Parkinson's disease and some of them to Alzheimer's disease. And why is that important? Because we can look to those research and care fields, and we can draw from them and learn from them and maybe take things from them that are relevant to our rare disease. And MPAN, more than any of the other NBIAs, has some parallels with Parkinson's disease and some closely related disorders. So there's something called Lewy body dementia, which is, we sometimes call it a Parkinson's plus disorder. And the way it differs from straight Parkinson's disease is this glumped together protein that's called alpha-synuclein. Instead of being limited to one part of the brain, it is all over the brain, and eventually. And that is, when you look at the brain of somebody who has died with MPAN, it looks so much like Lewy body dementia, but much worse. And, you know, that is a little shocking, but it is also potentially advantageous. And why is it advantageous from the point of view of developing treatments? Because there are many, many more resources being directed towards Parkinson's disease research, towards understanding the biology of alpha-synuclein, which again is not the protein sort of at the root of MPAN, but it is the consequence of that protein's rogue actions, if you like. And so there are literally like thousands of researchers and millions and millions of dollars of research being poured into trying to solve the mysteries of Parkinson's disease and related disorders. And we, in the MPAN community, can sit here and watch that and see what we can take from it and how we can use it to our advantage. To give you one example, and I don't want to overstate this, but there is a clinical trial that is underway right now in Parkinson's disease that is looking at one of the first really hopeful anti-alpha-synuclein treatments. So this is a drug treatment that is targeting the glumped together bad protein in Parkinson's disease. And it's a multinational, international trial being run by a big drug company. And they're very driven and focused on Parkinson's disease, right? Because that's where the money is to be made if you're a drug company, because there's lots of people walking around with Parkinson's disease. But it's a very exciting prospect for Parkinson's disease because it is going after that glumped protein. I'm sitting here in the MPAN world, like watching that and thinking to myself, you know, if that pans out for Parkinson's disease, we've got Parkinson's disease in spades in MPAN, and we might be able to hook a drug company after they've proved their case in Parkinson's disease. Then drug companies are very interested in having what we call additional indications, and that means another disease that they can go to the FDA for and say, hey, we want you to offer additional protections for our drug for this rare disease. And it is another market for them. Now, the disadvantage is in the US, there's not a ton of MPAN patients. You know, of the rare diseases, you guys are a little rarer than some. There's a whole lot more BPAN, people with BPAN floating around than there are MPAN in the States. In Poland, you know, as a percentage of the population, there's a whole lot more. So it might be a bit of a hard sell for a drug company to get them interested in an ultra-rare disease like MPAN. But the way in which we might do that is because you could potentially, I don't know, think about a study where you could get biological samples during the course of a clinical trial that have a greater chance of showing what they want to show. I don't know if that makes any sense to anybody here, but in this clinical trial in Parkinson's disease, for instance, they are taking samples during the treatment trial of spinal fluid because they can measure the glumped protein in the spinal fluid, and they want to see, is their drug, you know, before treatment and then at 2 points during treatment, does the glumped together protein level change? Because that then says to them, our drug is doing what we want it to do. And in MPAN, it would be pretty challenging, but you could think about a clinical trial where you could potentially get some spinal fluid samples during a treatment trial. So, you know, treatments, that isn't going to the root cause of MPAN, but we can learn a lot. We can benefit from what is going on in Parkinson's disease particularly, because it has some remarkable, important similarities to Parkinson's disease. So I want to leave you with a hopeful thought there. That's probably long-winded, but I think it's a good thing. Yeah.
Yeah. Question? I was going to bring up Lewy body dementia. I do believe I read your paper, because you have had a paper published about MPAN. Well, yeah, it was a while, because I knew about Lewy body dementia, being a nurse, and I knew about it, but I was surprised when I read the paper, as I say, it might have been an old one, that it was mentioned with MPAN. I was surprised. Now, my curiosity is, do the other NBIA subtypes have the Lewy body dementia, or only the MPAN? Or maybe you can't answer that. Oh, okay.
Yeah,
I
so
was curious.
so in Parkinson disease, the synuclein and the Lewy bodies sit in one part of the brain called the substantia nigra.
Can I just give one clarification
Right.
as to term? So when we say Lewy body,
Right.
so Susan's talked about the glumped together protein. There's a lot of names here. So the rogue, the protein that glumps together is called alpha-synuclein. And when it glumps together, what you see under the microscope, if you're a pathologist, is a glumped together thing that we call a Lewy body, just the terminology. And so, you know, a lot of alpha-synuclein glumped together equals Lewy body, just so you know the words, because we're throwing them around here. Sorry, carry on.
Yeah, so those Lewy bodies are limited to the substantia nigra. In Lewy body dementia, they're everywhere, or at least certainly more widespread. In MPAN, they're everywhere and 10x. They're far.
Now, isn't that what, what's his name, the comedian? Yeah, he had Lewy body dementia.
Yeah,
I mean, they
yeah.
didn't know it probably until post-mortem, right?
Yeah.
Exactly.
So synuclein is the dominant protein problem in MPAN. Synuclein is not present in PCAN,
Okay.
but it is present, and it probably is quite relevant in PLAN.
Okay.
But PLAN also has, so PLAN has tau and synuclein. And I think in more sort of more of a balance, and then BPAN is tau dominant with a little bit of synuclein sprinkled in there. Yeah, and
There will be a test.
yeah. So I mean, we do kind of I kind of think of them as, you know, BPAN. PLAN is, I think, in between, and MPAN, I think of them as a spectrum with sort of tau going up and down, synuclein going up and down. I don't remember if I think there's a little bit of tau in MPAN, I don't actually remember. And then PCAN stands alone because neither of those proteins or ApoE accumulates in PCAN neurons, but ApoE accumulates in PCAN neurons in all of the NBIAs. Yeah.
I guess what I'm curious about, because I've been involved and seen different families, et cetera, is dementia involved in all of MBIA or only in some of MBIA? Because my kids definitely had it. I didn't realize it at the time, but I look back and I'm like, wow, yeah, a 20-year-old with dementia, yeah, you don't usually see that. So I guess I'm kind of curious, which obviously it does affect their cognitive abilities, are maybe like the PCAN kids, maybe their cognitive abilities are a little better, maybe they don't have the dementia. I guess I was curious, do you think the Lewy bodies contribute to the dementia? Or? Yeah.
Because when we say that,
Yep.
when we say in PCAN, the problem's
Right.
not synuclein, but it's limited to one little part of the brain, and the rest of the brain really looks pretty healthy.
Right.
In MPAN,
Right.
the alpha-synuclein, these glumped together Lewy bodies, is all over the thinking part of the brain, what we call the cortex.
Right. Okay.
So the thinking part of the brain is what you got to have intact to not have
Right.
cognitive problems.
Right.
Now,
Okay.
dementia is not dementia is not dementia. There are
Right.
different flavors of
Right.
dementia. And, you know, as you talk to somebody who's had some of our adults that are here at the meeting who've had PCAN for a long time, they're a little off the wall sometimes.
Oh, yeah.
But you wouldn't. but
But
they're not
cognitive ability.
demented. They have, you know, there's a few things that might not like totally line up all the time in the way we might think of somebody the same age who didn't have PCAN, but it's pretty different experience than interacting with somebody who's had MPAN for 30 years, which is not to say anything at all about, you know, the wonderful people that you all have in your lives, but it is you have seen, all of you, because you all have older children with MPAN, you have seen big changes in their cognition over time, and that's not true for all the NBIA disorders.
Right. Yeah, I didn't think so.
And go ahead, James.
So the study that's going on for Parkinson's, so they can't cross like international lines because Poland and Germany have so many people with the MPAN and
the
that?
study
If
that's
they
going
could-
on in Parkinson's is international. Yeah.
Okay, but like, so to get them interested, there needs to be a financial gain. And so to do that gain is much more international borders. The other side, are they looking at that as what one of the questions I'm asking? Does that motivate them, I guess?
Yeah, it may well. I mean, even if there's not so many MPAN patients in, no, we're not there yet. So, you know, they've never heard of MPAN at this point. But even though there's not so many MPAN patients, certainly by population numbers in the U.S., there's a tiny fraction by population in the U.S. than there are in Poland. The reason a drug company would be interested to try to make it work in the States, even with an ultra rare disease, is because it then puts it in front of the FDA instead of the European drug authorities. And it is important for a drug to be approved by both the FDA equivalent in Europe, which is called the EMEA, and the FDA. And sometimes that, and I'm just talking like a drug company now, but sometimes that can be done with information purely from a European trial, but usually not. They want to see a study that is done in the US. So they might be interested in doing that. I just, as somebody who's done clinical trials, I'll tell you that the challenges of doing a clinical trial in MPAN patients in this country would be great. And it is because of the distances involved, the challenges of people who have MPAN to be able to get to a study center. But, you know, I guess my point in mentioning that drug in particular was really to, because I don't want to overstate how close we are to anybody pursuing that, but it is because there's so much effort and money being directed towards Parkinson's disease research, and more than any of the other NBIAs, MPAN stands to benefit from that. So that is the message of hope that I want you to take away, just from that sort of example, if you like. It is not to like dangle that particular drug as a possibility so much, but I mean, I think
I was just looking for a way
Yeah.
to motivate them.
Yeah,
You know?
right.
And if it means there are more people over there, hey, keep that in mind, guys.
Yeah,
You know?
yeah.
But the other part of the thing that I was wondering is so talking about this replacement and the glumping and all of that. So does that mean that that the replacement goes in there and actually the gene that's incomplete gets replaced, and then what happens to the incomplete gene? Because we're hoping that the good gene, right, the one that's intact, that's replacing, stays in there and does its thing and continues. But does that even, so there's multiple parts here, this mind is nuts. So there's, you know, then does it keep on regenerating?
Yeah, you're asking actually a really intriguing question. So in a recessive disease, like most of MPAN, right? Not all of MPAN. There are some families where only one copy of the MPAN gene is misspelled, and that's enough in those families. But let's not talk about them right now. Let's talk about recessive. Two copies misspelled, one from each parent. In that case, the nature of the misspelling, I think, so what with gene therapy does is generally not swap out the bad for the good or the good for the bad. It's not a swap in place. It just the gene comes into the cell and is integrated, but doesn't actually do a swap. There are ways to do a swap, and that may be where gene therapy goes, but generally the simplest way is not to do that swap, is just to put a new gene in and it gets sort of integrated and starts to behave like it should. So it's producing the functional MPAN protein and presumably having a positive effect on the health of the cells that get that gene. The story gets more complicated when we talk about dominant MPAN, and I can do that if you want. I can talk to you about that if you want. But that's a thought experiment, and since I think none of you has dominant MPAN in your families, I'm not positive about that, but I think that's a true statement. Okay, then that may be actually a challenge in dominant MPAN, because in dominant MPAN, the protein that is made doesn't just go away and not do its job. It actually takes the good one down with it, if you will. It sort of it joins up with the good one, and the cell kind of gets rid of it. And so if you're now putting more good copies in, good protein in, the question is, how much do you have to put in order to overcome the takedown process? And I don't know the answer to that, and that would lead me to think that a different approach, maybe the swapping approach or some kind of what's called gene editing, this CRISPR-Cas9 stuff you might have heard about. Some kind of gene editing might be actually more beneficial, where a chemical scissors goes in, snips the DNA, pulls out of its bag the right chemical to snip in, right? It's on a little chemical Pony Express going into the cell, little chemical scissors, little chemical bag of the right chemical. Yeah, it's complicated, and it's a very promising technology, but it's not ready for human use. Not at all ready for human use. Yeah. You never know. I said to one of the groups yesterday, I think it was the BPAN group, I said, I didn't think I would see successful gene therapy in my professional lifetime. Right. I'm 64, and we are seeing amazing results from really diseases that, are also devastating in the newborn period. Game-changing gene therapy, vision regained kind of gene therapy. And so never say never is super true about gene therapy. Yeah.
I was going to point out one thing that's sort of implied in everything that Susan and Penny said, but looking at the glumping and these different proteins we're talking about, we can only do that with donated brain tissue. And I think I've talked to all of you about this at different points, but there are questions that we can only answer with brain tissue with our neuropathologist in the lab. So families worked really hard to get us that brain tissue, and we worked really hard to get it right and fast and back to us. So it's just something to keep in mind, and something that I wanted to put out there for people who watch the recording later.
Yeah, and it seems to be a resource that isn't generally available it's not a practice in Europe. And so, yeah, and I don't know if that's universal in Europe. I don't know the challenges and what the barriers are, but we've talked to our German colleagues about this, and they seem not to have a path to obtaining human tissue, human postmortem tissue. This is true, but they were bad. They were bad guys.
Bad ones, I know. What?
Yeah, those people didn't exactly donate their brain tissue, did they? Yeah. 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 So, and in fact, that may be part of the barrier for human tissue. Nevertheless, what I just told you about the BPAN brain tissue, the tau dominant, we couldn't have known otherwise. This is a very new discovery, weeks old. Yeah. And the MPAN community, family community in the U.S. has been extraordinary in their gifting of brains. We take every care to handle those in very respectful ways and to honor how precious that tissue is. We also make that tissue available to the larger research community. We have a collaboration going with a group in Germany that's interested in how synuclein behaves and how the sort of brainstem behaves in one of the other NBIA disorders, but maybe she would be kind of interested in jumping on the MPAN bandwagon. Anyway, it's an exceptional resource. And I know some of you are already on board with that.
So like the fruit fly, can you also, do that with the brain tissue? Uh-oh, I stumped you.
Yeah.
Well, you can, introduce, I guess, NBA, the effective gene into the fruit fly. Can you, do that with the brain tissue and not necessarily have a person that has, I didn't mean the brain of a fruit
yeah,
fly.
you mean can we do it with...
So say you take my brain and you take that tissue and you introduce NBIA into it, whatever version, and is that a feasible thing?
Yeah, after the tissue has died, it's not I mean, after a person has died and we have gotten brain tissue, it's not really possible to introduce the gene and expect to see any kind of change, because we're looking for change in how the cells function. And if the cells are dead, by definition, there's not a function to be recovered. We can take skin cells. Many of you know we're doing sampling at the other end of the building, including skin cells. We can take skin cells and turn them into brain cells, and then we can infect them with, we can put a new gene in those. We can put a bad gene in and a good gene. We can do all sorts of gene therapy experiments. And that work is being done. I think you'll hear more about that by Dr. Ayuso tomorrow.
So we need to go down there and do that? Is that something we can do?
Yeah, we sort of have put some framework around what type of samples we need from what kinds of families, and so I don't know off the top of my head. Probably not skin cells from you, but if you want to contribute a blood sample, I think we would be very appreciative.
Okay.
Totally up to you. Totally discretionary.
You just got to lead the way.
But it turns out that skin cells, once you get much over 35, they don't grow so well.
Okay.
So we have devoted a lot of laboratory resources obtaining skin samples and then trying to grow in the lab skin cells from people who are over the age of 35, and the young'uns is what we need. So I hate to break it to you, but most of us in the room are not much use. I'm looking at you, Lakin.
You do have some clinicians in the room, and even though they're pediatric people, adult person, I would encourage you to talk to them if you have medication questions, management questions, or other questions to ask in terms of helping your family members.
I'm curious if you need the whole brain, or like you take a piece of the brain for after they have passed.
Yeah. So what's generally most helpful to us is whole brain. Yeah.
But I will say, too, we've had a really good experience, and the people who collect that tissue know what they're doing and do it in a respectful way, where you could even have an open casket typically afterwards. It's not what you're imagining, or what I imagine you're imagining. I know it sounds a little scary, but yeah.
So for the other clinicians in the room, medications for MPAN patients? Now, I know my kids have tried baclofen. I'm not sure that was, well, the baclofen pump did help Bruce, but as like me and Barbara, she's 42, and I'm even thinking of Gigi a little bit. Is there anything other than Botox with some of the muscle contractures, or is there anything else that's suggestible medication-wise that would be helpful? I mean, Barb's happy. I'm happy that Barb's happy, and she doesn't have a trach or a feeding tube, but so we stopped at a certain point. Baclofen really wasn't helping Becky. Bruce had already passed. And we stopped it with Barb because I really didn't see any difference. So I don't think it was doing anything. When Becky was younger, they did have her on Eldepril. I even forget what that is, but it did seem to help for a little bit. But I just didn't know if there was any other suggestion medication-wise.
I think, the important thing is just to look at what the symptoms are that may be bothersome.
Right.
And I agree with you. There's oftentimes that, loved ones get put on medication, but you did the right thing. Like, is it really making a difference? Right. And that's one of the things I encourage. Yeah, I don't. Right. And you took her off. And if you don't feel like you noticed a big difference in kind of some of the movement-related issues that you can have, then I think it's reasonable to minimize medicines that you think aren't helping. I think the hard thing is that for everybody, it's a little bit different as to kind of what is impacting them. So I think it's really individualizing it to what their needs are. And I always tell my families, you know, you may be on a list of medicines. Keep asking me. I need to ask myself, do we need to be on this or not?
Right.
And that's really important, especially in this long journey. They may be put on medicines long ago and it's just been on there and
Right.
no one knows whether it's helping, it's not helping. You can have side effects. So I think you did the right thing.
Yeah.
Hi, good question for the movement disorders people. Our daughter's 23, and for the last 3 or so years, she was having more and more dystonias, mostly in the lower extremities. She had Botox done twice, and that did soften up the dystonias, but then she couldn't stand well, so we're not doing that anymore. In the last 8 months, as she's progressed more rapidly, she has both truncal and neck dystonias. And it's been discussed with our own neurologist doing a Botox block for those. So my question, what's been the experience with that? Is that likely to be somewhat successful in
I think
managing
Penny,
that?
yeah. I can start and then Penny if you want. I think it's important. I think the nice thing about Botox is it can be targeted, which oftentimes the medications that we have that we use from a tone perspective can get tricky because you end up running into side effect issues. I think most times it's not you're right. I think she will likely have some benefit from it. That's the goal. It's probably not going to do everything that we want it to do. But ultimately the goal is that if you're able to use the Botox and give some relief, you can then look at kind of her current medication regimen and see if there's some adjustments that you can make there that may be helpful. I always say, look at the medicines you're on, see what medicines we can adjust rather than adding more medicines on. And then the timing of medicine is the other thing I've done with a lot of families. Look at the time that the medications are being given, because oftentimes we do it in sort of at the same time, and I've had some luck with some staggering of medications that I know can be hard in trying to give the medicine, but sometimes it helps in lasting of some of the symptomatic relief that patients can have. She's passing it to you.
Okay. Yeah, no, I'm just learning a lot from you. Yeah, I think with injections, I think it's okay to try and see. I mean, that the upside and the downside is that it's a temporary treatment, right? So, I mean, there are risks, obviously, with doing any injections, and especially if you're doing around the head and neck. But I think with a experienced provider, then, you know, it might be worth a try if it's a bothersome enough symptom. So.
You can sort of look at what the twisting movement is, and sometimes, you know, very effectively target one muscle and quiet it down. I think the things we worry about with head and neck injections are, depending on which muscles are involved, you want to try to avoid getting into the swallowing muscles, because then that can cause problems. And those of you that have sort of been around that block, you know, that's where an experienced clinician, you know, they're going to proceed very carefully when they know that they're injecting a muscle that is kind of close to the swallowing muscles. They'll maybe start at a lower dose than they think they need, and you know, it's kind of a long-term commitment. It's like, well, that helped a little bit, not quite enough, but it didn't cause any swallowing problems. So now we're going to, 3 months from now, we'll creep up the dose a bit more. That's the kind of thing. And, you know, some types of jaw movements can be helpful, but again, you've got to be super careful.
So my daughter has both truncal and neck dystonias. Would you go after one of them first and see what happens? Would you do both at the same time, or how would you strategize that?
I personally wouldn't treat truncal dystonia with botulinum toxin.
Not truncal.
I haven't had luck with that myself, and I mean, you can comment too. Yeah, I mean, in kids, I've seen actually levodopa work okay for truncal dystonia. I don't have experience with that in MPAN, so I'll leave it to the experts. And then, whatever you could do with like positioning and PTs and OTs and that sort of a thing. But I don't know if you have more tricks.
No, I think, again, like looking at the medications too that she's on, I agree with you, Jenny, that I think truncal dystonia is a little bit tougher. So I think maybe looking at the medications, looking at the time, like if it's also, are there times that it fluctuates? What are there certain things that may be triggering it a little bit more? And then looking at the medications to see if maybe, one that I've been using, gabapentin has been one that's been very interesting for me because there's a pain component to a lot of dystonia as well, that I think sometimes that can be helpful, but it is a little bit of trial and error.
So the program starts in the ballroom at 9:15.
Oh, it's 9:15.
But we should, wrap up.
Okay.
James had one more question. Not James, Michael, sorry.
So I just wanted to hit on one more thing, and that is that, Allison, you're amazing getting the information through from you guys. And we're very, very grateful. So I'm not, saying anything negative. But what I am saying is that, we talked the other day and brought up something that was really pretty interesting. So we were at a birthday party in Philadelphia and got a telephone call from Hattie Larlam. Jim's going in the emergency room. We beat Jim to the emergency room from Philadelphia. Good thing we did. And that's the reason I'm saying that. Because when we got there, the guy that was in charge of the ICU came to us and said, so what's going on with Jim? I said, he's getting Botox in his arm, so he's not going to do that finger thing. And I said, he has optic nerve atrophy, so he's not going to see the hand waving in front of you. I said, but he has really good hearing. And I addressed the other things that they're going to do the test on, right? So what does he do? He leans over to Jim and he says, hiya, Jim. And I looked at this guy and I go, did you not? He takes his finger and sticks it in there. And nothing. And so all of these different things. So, there was this total lack of taking what we just explained and applying it. And the next thing is that we have, we made decisions based on that information. And so being the head of the ICU at the time Jim was admitted, he was the guy. We couldn't go to the next doctor who was making sense. So with that information said, we talked about different things about feeding tubes and about trachs and all of that stuff in the heat of the moment. And a hasty, maybe I shouldn't have said that, but a quick decision was made on our parts. And maybe that the people that are going through this behind us maybe would benefit from having more information about the things that may be in the future is all I'm trying to suggest to you guys.
Yeah.
And again, thank you guys for being so much, leading us in what we can maybe see.
Yeah. So on behalf of my entire profession, I'm sorry that happened. I really am. So there is an emergency preparedness talk. I think it's happening this morning. And it's not just like there's a fire, how do you get your family out? Part of that is, and I think actually the pediatrics community has led in this way of actually preparing, doing an emergency preparedness document for your family member, child or not child, meaning a young person or an older child, and sort of walking you through the things that you should be writing down and should be in the medical record and should be available to the care team, wherever the care team ends up being. I'm going to pass the mic here so that if you guys know more about it, I think we put reference in the PCAN best practice guideline, but
Yeah, I mean, I think there are easily accessible emergency plans, but I think the key is getting your practitioner who knows your child the best to help put that together with your input. And you have to just think about what are the things, like you just said, that the doctor needs to know when your child arrives in the emergency room, if you're not there, or even if you are there. And how do you highlight those so that they're as bold and big as possible? I mean, in truth, you did those things yourself. And so that was not the problem in that situation. It was something else. But I see.
So he could have a, I mean, that's pretty simple, it's analog, right? MPAN, you know, poor, low vision, you know, I mean, you could just put something analog on them so that.
Like the top 3 things you want somebody to know in 6 words, you know?
They're also high-teching this stuff with like QR codes. So put a QR code on the bracelet.
It's crazy because you know it's like the so it's like the person that's in charge of finances for the doctor. She says, Mike, you just need to call them and tell them that you're not responsible for that medical thing, you know, the hospital. I went, really? I could do something like that? Why would they listen to me? They're not going to listen to me and say that I'm not responsible for a medical procedure. So I wouldn't even, you know, I don't know even how to go to begin that. You know, maybe I'm naive. I don't know, but.
We'll talk separately. I want to honor.
We're starting announcements.
Yeah, we probably should.
Thank you all.
Yeah. And we're around, you know, through to Sunday, so find us and talk to us about your individual family members. Sorry, Jenny. Oh, no, that's okay. I got your list, Jim. Put it in my pocket. Keep saying Jim.
I know, no, well, I started it.
Started it.
He's in my mind, yeah.
Oh my